Okay, so our next speaker this afternoon is Dr. Pablo Pineyro, and he's an assistant professor in the Department of Veterinary Diagnostic and Production Animal Medicine at Iowa State University. He's also a diagnostic pathologist at ISU Diagnostic Lab, and his research focuses on infectious diseases of swine and their associated pathophysiology, and also diagnostics. This afternoon, he's going to talk about an update on a few viruses in swine production. So join me in welcoming Dr. Pineyro.
All right. I'll give you a few seconds so people can move in and out. Okay, thank you for staying here this afternoon. I work in the Veterinary Diagnostic Lab here in Iowa, as most of you probably know. We are one of the busiest diagnostic lab associated with food animals in the United States. So we handle about 75,000 cases a year, and about 80% of them are pigs. So we get to see quite a bit of diseases going through our daily work. So today I'm going to just touch bases in some of the things that we currently see in these three diseases up here: PEDV, PURRS, and Seneca virus. So first, we're gonna kind of review what happened with PEDV during the last few years and show you where we are now. So just a reminder, what is PEDV? PEDV is a virus that belongs to this family, the coronavirus, Coronaviridae, and I'm gonna just show you 3-4 things that we're gonna use later on during the talk for the diagnostic side. So the structure of the virus, is this thing working? Okay, the structure of the virus is the coronavirus means that they have kind of a crown around, and that is the typical morphology of the family. So the virus itself has open reading frames, so these open reading frames are going to codify for structural and non-structural proteins. And the ones that we're interested in are these 4, the spike protein, membrane, envelope, and nucleoprotein. Those are the structural ones. And I won't probably remember those because we're going to use them when we talk about diagnostic. So all this coronavirus family is divided in 4 different genus: alpha coronavirus, beta coronavirus, gamma, and delta. And it seems like all of them have like a common ancestor that came from a bad coronavirus, the sham-tooth birds, and they were spread to the different mammalian species. But what is important for us? Which ones are the ones that affect pigs? So pigs get affected with multiple viruses within this big family. So instead, in the alpha coronavirus genome, we have 3 viruses that affect PRRS: the PEDV, porcine epidemic diarrhea virus, the porcine transmissible gastroenteritis, the TGE, that we don't see that often anymore, and porcine respiratory coronavirus, which is kind of a mutation of the TGE. So all those 3 affect pigs. Then the beta coronavirus, porcine hemagglutinin encephalomyopathy, we don't see that quite often, but it's also another disease that can be caused by coronavirus in pigs. And finally, delta coronavirus. So you can see we have 3 genus represented affecting the swine population. But I like to kind of reorganize this. That is within the genome, species, but I like to see where these viruses and what is the clinical presentation that they have. I like to organize this in this way. So enteric viruses within this family: porcine endemic diarrhea, TGE, and delta coronavirus. Those are the ones that are going to cause diarrhea in neonatal pigs. Respiratory, porcine respiratory coronavirus. The clinical presentation is not as severe as any of the other respiratory pathogens that we know, but it still can cause disease. And this one that I mentioned before, the PHEV, basically is a CNS virus. It causes necrosis of the neurons, but what we see is a gastroenteric clinical presentation. Basically, all the myenteric places get paralyzed, and those pigs vomit a lot. So it used to be the disease used to be called vomit and wasting disease. So, but it's not because the virus affects the GI tract. It's because it causes a neurological problem. So what is the situation of PEDV? That's the topic here. So as you can see, it's a kind of random slide here, and this one tried to represent where we are now. So with the epidemiology, diagnostic tools, and the genetic of the virus. So just a brief history. Let's review what happened in the last few years. So back in 2013, actually I was working in Virginia, way far from the Midwest, and we got this peak. Actually, that picture was taken with my cell phone there, and we got this diarrhea with this thin wall intestine, translucent, all filled by gas, kind of atrophic enteritis. We ran the common pathogens that we knew, TGE, coronavirus, rotavirus, and all of them were negative. So we sent the sample for sequencing, and we found this virus close related with the PEDV that had been reported in China. At the same time, here in Iowa, people in the diagnostic lab, they got about the same picture. If you look at the intestine, kind of look the same, this translucent, really thin wall intestine, and the histological picture is the same. So, and they came to the same conclusion. They did a sequence, and they did the first diagnosis of PEDV. So this is how everything came up. But here is kind of a time lapse, how things progress. So the first cases were reported late April, early May in 2013. Histologically, again, we see this atrophic enteritis that resembles like any other enteric virus pathogen. So TGE, rotavirus could be, but by PCR they both were negative. Remember that I talked about this coronavirus structure. So we did electron microscopy, and here we are. We got the first evidence that the virus was affecting pigs. We ran a pan-coronavirus PCR. Remember that I mentioned all these genomes, the alpha, delta. So these tell us that it's a coronavirus. Doesn't tell us which one it is within those genomes, but it's still telling that it's a coronavirus. So we weren't getting close. We did a full sequence in conjunction with NBSL, and we found that it was a close homology with a strain of PEDV that was reported in China in 2012. So by mid-May, not even a month later, USDA came with the first official report of PEDV in the United States. And about a month later, we already have a PCR in place to diagnose. Remember that I mentioned those genes at the beginning of the talk? So our PCR was based in the spike gene. That is what was important, the anatomy of the virus. So, but this, as you can see, is just less than a month progression. After this, just the wheels came off and everything was a disaster. From there, we got this massive diarrhea, pigs dying every day, reaching 100% mortality in most of the farms. And our lab in the lab went really, really crazy. But from those first cases in 2013, what happened today? What is the situation today? So USDA came out with a weekly update of PEDV cases. And look at the pig producing states, Iowa, Minnesota, North Carolina, Kansas. So in Iowa, we have 1,300 premises reported in the last 3 years. If we think about farms that can reach 100% mortality, think about how many pigs we have lost in the last 3 years. 13 premises with 100% mortality. That's a big number. Another thing that we learned from the epidemiology is just 3 years data. So look at this. This is a weekly report. We have a seasonal pattern. So disease is affecting more the winter and is giving us a break during the summertime. Perhaps the virus can just resist the hot weather, and that is where we get a break. But it doesn't go away. Even when the scene that is clearing up during the summer, it doesn't go away. Cases came back again, and we have rebreaks of PEDV every year. So that is kind of the epidemiology and the situation, the current situation. Now, I want to show you probably what it was the first experimental reproduction of the disease that was done here in Iowa State. So CDC pigs, so cesarean-derived, calostro-deprived, they don't have any immunity at all, they don't have any protection against this virus. They were infected orogastrically with a really low dose, 10 to the 3. And we took samples fecal swabs, we did necropsy, a time course necropsy, really short because the duration of the clinical signs is really short, no more than 3-4 days. And we necropsy and evaluate what region of the GI is the most affected one. So 12 hours post-infection, 30% of the animals were already showing clinical signs. They already have diarrhea, dehydration, and by 24 over 72 hours, 100% of them were affected. So vomiting, diarrhea, and we start seeing mortality there. So this is probably the first time someone really got the virus, put it in a pig, and reproduced the disease. Here, just for compare, I know that I already showed this picture before, but this is how a normal gut will look like. A really thick wall, we can see the vessels, the hyperemic vessels. Compared with this one, it's really thin, translucent, almost empty. So that is what we normally see in a PEDV affected animal. How this looks histologically is how we start making the diagnosis. And this is going to also help us to understand the pathogenesis. If you think this is a picture of a normal intestine, think about the normal intestine like fingers. So the piglets need all that surface to absorb the nutrients. If that surface gets affected, we start getting reduced, on top of diarrhea, the reduction of the surface that is going to absorb the nutrients. So keep in mind this one. The virus can affect the tip of the villi here and the crypt. So these green cells are affected cells. So down in the crypt and top of the villi. So here's where we start getting necrosis of the intestinal mucosa. Again, the brown cells, affected cells, all the way down to the crypt and up to the top of the villi. And if you compare this one, we normally say that the healthy gut should have a ratio of 7 to 1. 7 along the villi compared with 1 to the crypt. Here they are getting really, really short. We are still losing that absorptive surface of the gut. Finally, we have what we call atrophy. So if you compare the length of this one, this is a severe affected one, with the normal gut. So we don't have pretty much surface to get any absorbed. Even if these pigs survive, they're not going to have a healthy gut to absorb any nutrient at all. So we have this atrophy, fusion, and blunting. So the intestine tries to repair the damage that the virus caused, but it's not going to be really effective and never going to get back to this probably original length. That is kind of the progression of the disease and the pathogenesis. It's how things happen. So the clinical signs that we see in naturally infected animals, we mentioned that before, diarrhea and vomiting. And it's going to vary a little bit based on the age of the pigs. The younger they get infected, up to a week, is really severe. If we go about 2 weeks, we guess the clinical signs are less severe. The same thing happened with mortality, about 100% in animals less than a week old, more than 10 days. It could be around 30–40%, depending on the immunity of the farm. So the time of incubation can go from 24 hours up to 4 days. And the clinical signs, as we saw in the experimental part, it showed really fast. And the other thing, if we had in a farm, the virus spread really quick. So that is why we have these high mortalities affecting pretty much every single pig. This is just a picture of how an infected crate looked like. So just one pig trying to eat. These are really lethargic, dehydrated, all covered by feces, and this is what we're normally going to see in a visit to a farm affected by PEDV. A really high mortality with mostly dehydrated pigs and covered by feces. What diagnostic tool we have? If you remember during my time course presentation, back in the day we only had PCR, and the morphology that was compatible with other diseases as well. So now we have a more complete toolbox for the diagnosis. So we have PCR, as I mentioned already. We have immunohistochemistry. This is a immunohistochemistry picture. So that is a direct method. It's going to show the virus in the gut. So there's no question about that. Serological tests, so indirect method to detect the presence of antibody if the pigs were in contact, but doesn't tell us the presence of the virus. So IFA, ELISA, virus neutralization, those are probably the most common used technique. So I put here no virus isolation. It's not because it not can be done. Actually, it can be done. The problem is take time. And when we have an outbreak like this, we need an answer right away. We can't wait for a week until the virus grows and we see the cytopathic effect. We can't wait for that. So that is why we don't use that one as a common diagnostic tool. And bioassays, that was probably one of the common tools that we used at the beginning. We get infectious material, we put it back in pig, and if the pig reproduces a clinical sign, that was our confirmation of the disease. Now we have other tools to make the diagnosis. So kind of the take-home for this part of the talk. So it causes severe malabsorptive and maldigestive diarrhea. Remember that I mentioned they don't have more villi to do the absorption. So that is a malabsorptive diarrhea. So it's acute, as I mentioned, in few hours we have the disease. So that is why I highlight acute and high mortality. Those are the main key features of the clinical presentation. Severe atrophic enteritis, I show you the slide how it's going to look like, and that can go for affecting just one portion at the beginning of the disease, duodeno and jejunum, the cranial portion, and can get spread throughout the whole small intestine as the disease progresses. Probably the most important thing about this disease, besides all this pathogenesis and how it looks like, is the economic impact. And as was mentioned in the previous talk, all these diseases, they have a face, and we have to remember that each one of these farms that were affected, it's not only the loss of the pig, there are all other implications, and the economic implication is huge. If we look at 300 million a year just for PEDV, it's an enormous loss. But it's still not compared with this. So, as you mentioned before, this is still really, really important. PURRS is still probably being our most important economical disease. That is why the next part of the talk is going to be about PURRS, what we see in the diagnostic lab, and what is required from us as diagnosticians. This is just kind of a summary of respiratory cases. As we know, I'm going to show a few pictures later. Respiratory PURRS came in different flavors. So respiratory disease, reproductive disease. I'm going to focus mainly in respiratory problems. So this is a percentage of cases, but different pathogens by itself. I don't even try to touch bases in co-infection. This is just pure infection. So PURRS is still being probably the most important. 40% of cases, almost 40% of cases are associated with PURRS, followed by SIV, Mycoplasma pneumonia, Pasteurella multocida, and other bacteria, and so on. But 40%, what does mean in numbers, in real numbers? So this is just total case numbers. Respiratory cases, diagnosis only with PURRS, nothing else. There's no co-infection. And we're talking about an average of 2,000 cases a year only from Iowa industry. This is what I was mentioning before, the different clinical presentations. So abortions, respiratory disease, and increased mortality in nursery and grower pigs. What is the current status? Hopefully, we're going to get the knockout pig soon, but we still have to deal with this. So this is how we battle the disease. We have vaccines. In our market, we deal with this, with the modified live attenuated vaccine. And as was mentioned in the previous talk, this vaccine helps, but doesn't prevent totally the disease. This vaccine is still getting animals sick. So depend of the status of the herd could be just a mild transitory disease, and we can see actually severe outbreaks only just for the presence of the vaccines. In Europe, they use the kill vaccine. The problem with that one is that the immunogenicity is not even comparable with the modified live. So the protection is not enough to prevent an outbreak of the disease. There are other experimental vaccines out there, viral vector, DNA, subunit vaccines, all of them to be proved to be have some efficacy somehow, but they don't work as well as the modified live. And unfortunately, it's probably the only tool beside management that we have to prevent the disease now. So diagnosis, this is what we do in the lab every day. So believe me, we still rely on this. We need someone to tell us that we have a respiratory problem. If you send us just a piece of lung and said, test it for pathogens, we can't help. Sometimes we need a little description, percentage of animal affected, what is the clinical presentation. Respiratory means a lot of things. Means coughing, means distress, means sneezing. So when we get our samples, we need a little more help to give you a better answer. So in this case, we have the typical blue ear that is how the disease used to be called before. This animal has cyanosis, they can't breathe, and they can't oxygenate and start getting purple. So this is probably the first clinical indication of respiratory problem caused by PURRS. Then when we open up the lungs, the pigs, we see these lungs that fail to collapse, they are heavy and wet. That is kind of indication of a viral infection. It doesn't tell us that it's PURRS, it's still a virus infection. PCV can also cause kind of the same type of pattern, so it still helps, but it's not definitive. We is what I normally do, is what I do most of my time, pathology. So this is what we look for in a slide. This interstitial pneumonia. Look at the thick alveoli. This will be just one single layer cell. And here we have really thick alveoli. This will be the air space. So if we look at the next picture, there's no air space. So it's gone. They don't have space even to breathe. And the confirmatory probably for us is this one. This is immunohistochemistry, and the brown dots in there are infected cells with PURRS. So they're infected macrophages with PURRS. So that is kind of the easy and faster way to approach the diagnosis. But we have more tools. So we have virus isolation. We can do sequencing out of the virus isolation that help us to confirm the diagnosis. Of course, PCR, and we don't only use regular sample like tissues and serum, we also use oral fluids. We need to not only do the final diagnosis of a respiratory problem, but we need to do epidemiological evaluation. We need to know if the virus is circulating. So this is probably one of the newest technologies, use oral fluids for PCR and get a sense of the dynamic of the virus in a population. Indirect method, I mentioned before, and I like to scratch this one because indirect methods used to be called just serological test. I like to say that it's an immunological test because now we don't use serum anymore. We use more platforms to evaluate the immunological response. Instead, we use oral fluids. So that is why it's not a serological test anymore. It's an immunological test applied in a different platforms, in serum and oral fluid. We can use that one for ELISA, immunofluorescence, and virus neutralization, and we can still detect antibodies produced by the virus. So these techniques are going to tell us the presence of the virus. Here in this picture, we just see a pen with a rope hanging there where we're going to collect the oral fluid, and we want to evaluate the dynamic of the virus in this population. Another question that we get frequently in the lab, what is the pattern? Clinicians, they need more information, not just the presence of the virus. They need more to battle this disease. So this is a technique that has been going on for a while. So this is a restriction fragment, polymorphism pattern. Basically, we put the virus with different enzymes, and the enzymes are going to cut the virus. They cut in little pieces. So what we see here, this shell are the pieces, different size and different weights of the virus. So what happened is different viruses that got caught in different sections. So and we start naming them by this fragment. So different restriction patterns. For example, the one that we use to differentiate the modified live vaccine is this typical pattern, 252. This is the one that we see with vaccinated pigs. Why I made all that explanation? Because this is what's happening now. We are seeing a shift in this pattern. So we typically see for quite a long time the 1-3 pattern. So these blue bars, look at this case number here. And now we are seeing more than 174 pattern. And this seems to be more pathogenic. When farms get hit with this virus, with this pattern, the clinical signs are more severe. So that is why we need that information we need to provide that information to clinicians. They need to know if they have this severe disease because it's a new virus, because it's a new pattern, so they need that information. They also need this information. When we get this piece of lung that we run a PCR and it's positive, well, but my pigs were vaccinated. And as I mentioned before, pigs were vaccinated with a live virus, so we can't tell apart. So, and that is another piece of information that we need to provide. So we have a specific PCR that can detect in this specific region in the OR5. We can tell the homology if we sequence and we do PCR with the known vaccine, Fostera, Inselvac, Primerpac. So in this case, the homology is about 80%. We can certainly say that is wild type strain. But sometimes this homology is really, really close. And we cannot tell. We cannot tell. So that is why maybe this is going to be the future. Do the next generation sequence. So the full length of the virus, just not that little piece of the OR5. We need to sequence the whole virus and see if the mutations or the difference are anywhere else more than the OR5. So probably those are the answers that we need to, or the question that we have every day in the diagnostic lab and we need to answer to the diagnosticians. Finally, Seneca virus. I believe that everyone has heard about this disease in the last few years. But what is Seneca virus? So basically, it's a swine vesicular disease. So if someone tells you swine vesicular disease here in a farm, what's probably going to be the most important disease that we're going to think about it? What is our worst fear? FMD. Yeah, FMD. So FMD probably is going to be in our differential for vesicular diseases, but it's not the only one. So we have swine vesicular disease, we have vesicular stomatitis, and we have vesicular exanthema of swine. So all of them, some of them within the same family of Seneca virus, some others are not. But how they look all the same. And that's probably the most scary part of the thing. When we got to the farm and we see these vesicles, so until we don't get any lab confirmation, we can tell otherwise. I mean, there's epidemiology behind, they behave differently, but the lesion itself, it can be tell apart. It can. So I like to keep this picture because with this picture up here is how everything started 2 years ago. I was on call that week, and a producer sent me this picture. We have this animal. This is a show pig in Wright County, could be, am I right? Wright County. So it came from a county fair in Wright County, and they have these vesicles up here, kind of blanching in the coronary band, hyperemia here, and that was it. So, and that pig was ready to move to another county fair. So we kind of suggest, why you don't keep it for a while and to see what's going on. So we got a swab from these vesicles. Of course, we sent it to MBSL to test for foot and mouth disease. Thanks God, we ruled it out. And we knew that by the time there was an outbreak of Seneca virus in Brazil. Brazil, like a month before, they have reported an outbreak of vesicular disease and neonatal mortality. That is why I have that picture in here, to remind us that they have two presentations, the vesicular presentation and the neonatal mortality. So knowing that that was going on in Brazil, we requested to test for Seneca virus and turned out that this one was positive. So these animals here were the first animals that were diagnosed here in Iowa State. So we start getting more and more cases. We have luck, and most of the producers help us keeping animals and allow us to do some tests to learn more about this because we didn't know much. So first thing is in a finisher farm in North Dakota, they sent us 5 finisher pigs with vesicles. So we opened those pigs and we checked head to tail everything, and we couldn't find any other lesion more than the vesicles. So we learned something, that nothing else more than a vesicular disease was going on. So if you see the lesions here, vesicle in the nostril, ulceration of the plantar region, this is an acute vesicle, again, ulceration of the plantar region. And believe me, if you walk into the farm 1 week after this outbreak, you won't find anything. The lesions have resolved and the animals are just fine. So this has to be detected in the really, really acute stage. This is the other presentation that I've mentioned, the neonatal mortality. So you go to a farm and you see these healthy litters here, and the next one is affected. So for a while, people being described and associated with this presentation diarrhea. So far, we couldn't make any cause association with the diarrhea. We detect the virus in pigs that have diarrhea, but when you try to identify the virus in the intestinal layer, they don't show up. So yeah, it's associated with diarrhea most of the time, but Seneca virus is the cause? I don't believe so far. This is the animal that I mentioned that we got from South Dakota. We have 5 finishers here in the diagnostic lab. We were able to check every single tissue. So if you look at the variety of lesions, just kind of hyperemia in the coronary band, more ulceration, blanching. Here is kind of ulcerated. It lost the hoof completely. Acute ulcer here, acute ulcer in the plantar region. So, for example, if you get this animal, no one goes through the farm just picking up every leg. So you will miss that one. And sows are all day laying down, so that they don't show lameness that easy. So that is why it's so sometimes the clinical diagnosis is so hard to make. This is the the nostril. Remember the picture that I showed you with the different vesicular diseases? This one doesn't look any different with the other ones. This is what happened. This one was like 10 days post-break. So it was already repaired. This is scar tissue. So you will barely going to see this lesion. So the timing of detection is really important for the clinical diagnosis. Here's another one, scar tissue, a little bit of hyperemia, but nothing else. In this farm, so the owner helped us, and he kept 10 pigs for 3 weeks when those guys were ready to go to market. So we keep those animals, and we didn't know much. So we tried to learn as much as we could and as fast as we could. So we took serum for 3 weeks. Here I should say they post outbreaks because we don't really know when they were infected. So there's no day post-infection. They just break. 36 days later, we start sampling them every week. So what we learned here, first, the serum. There was no detection. To me, this is a totally outlier. And even, let me mention this. Can you see there are 2 lines in the CT score? Because we didn't even know which was the cutoff. So these are the first results. I mean, we ran everything up to 40 cycles, but we didn't know which one was the cutoff. So serum was no detection, which means for us that the viremia didn't last that long. So we were 35 days in post clinical outbreak, and there was no virus detected. Nasal swab, rectal swab, and tonsil swab, I kind of try to understand that as a shedding. So by oral route or fecal route, so we still see animals positive at least 36, 43 days post-break. So it means for us that the virus is still being shed for several weeks after the clinical presentation. So for the diagnosis of the disease, when we just started with this, we have only the PCR, we did a full-length sequence, and there was a variation in this virus from the previous reported. I mean, this virus had been around for a while. It's not a new virus. The difference is that we haven't seen clinical presentation, but the original virus, the Seneca Valley virus, the SBV, so we compared our sequence with the original SBV. Let me see, here is. And it was about 10% homology if we look at the BP1, which is the main structural protein, and 3% if we look at the whole genome. So there was a little bit of difference, but we didn't know if the clinical presentation was immune evasion of these new strains or just happened, really. Another diagnostic tool that we developed really quick to make the diagnosis was in situ hybridization and immunohistochemistry. This is an immunohistochemistry. So basically, we are labeling the virus in this vesicle section. So we cut through the skin, put the antibody there, and we detect the virus. Another thing that we learned from that clinical experience, so we were testing basically vesicle fluids, but is any other tissue useful for diagnosis? We didn't know that. So those pigs that we necropsy and check every single organ, of course, we PCR every single organ and see if we can come out with a consensus, with a pattern, which one would be the best sample. And we found virus all over the place, but consistently, the vesicles were, of course, the best sample. Inguinal lymph node was useful, and tonsil basically give us the option to use oral fluid. If it was in the tonsil, perhaps it's going to be in the oral fluids. So again, the techniques that we've been using, so we developed immunohistochemistry, as I mentioned before, in situ hybridization. Here, instead of using an antibody, we use a nucleotide probe that binds to the virus. It's a little more specific. Virus isolation. Here we got a nice monolayer of porcine testicular cells, and here is an infected one. So we have a really strong cytopathic effect in 24, 48 hours. This is an electron microscopy to confirm the diagnosis. So those were the first techniques, basically techniques that have been used for a while. We didn't develop anything original, but we need those tools to make the diagnosis. Now on, we have indirect methods to make the diagnosis. We have ELISA, we have developed an ELISA based on targeting the BP1, this protein, this structural protein that I mentioned before, and we also using IFA. Here is a picture of the IFA, and we also have virus neutralization. So those are the indirect methods that we use for antibody diagnosis. A little bit of neonatal mortality associated with Seneca virus. As I mentioned before, people have described this diarrhea and just mortality, but we couldn't find a cause association. And actually, this is a case where we've been called because of this. There was an increment of mortality in that week. If you look at the numbers, I mean, that's not really spectacular. So nothing like PEDV. We have a slime diarrhea, an increase in mortality, but we don't have to really worry about it like PEDV in the enteric side of the disease. Mostly we see associated with E. coli or rotavirus, so who knows really if Seneca virus is the cause there. We did a little field investigation because what we saw is that this neonatal mortality was present, but there was not a real association with clinical affected sows or non-clinical affected sows. So what we did, we walked into the farm, we looked for animals that have vesicles, and we are ready to farrow an animal that didn't have vesicles. So we have 11 and 11, clinical and non-clinical affected, and we followed their offspring for a couple of weeks. So percentage of positive sows, in blue here, the clinically affected, and in red, the non-clinical affected. So at least we have half of the percentage affected animals that were positive to Seneca virus that didn't show any clinical sign. And when we look at their piglets, the same thing. So half of the piglets coming from south that didn't have clinical signs, they were positive for the virus. So we took this experiment a little farther, and we followed those animals for a period of 6 weeks. It's as long as we could go. So sows, again, we did 3 platforms: feces, tonsil swab, and serum. If we look at the serum in sows, we detect the virus for probably 2 weeks after the break. So again, this supports the original idea that viremia doesn't last really long. And now experimental inoculation have proved that the viremia doesn't last more than 10 days. If we go look at this group here, feces and tonsil swab, maybe we can get the same idea that we had before, just shedding of the virus. The virus gets shed for a little bit longer. Up to 6 weeks, we're still detecting the virus. Now we're going to move to the piglets. Look at the piglets. So there's virus in serum that decay 4 weeks after birth, and there's also virus elimination that also decay about 4, 5, 6 weeks, so basically up to the time of weaning. So these guys didn't get affected, but they were shedding virus. When we look at the antibody response, the sows were sampled 1 week post-break, and there's no different antibody response in clinically and non-clinically affected sows. So something is going on. The sows are getting infected, but they are not showing clinical disease. Or perhaps the caretakers are missing that clinical disease that we mentioned before, because it's so brief and so acute that we don't, if you don't check the sows really close, we miss the clinical presentation. And the piglets, they have a decay in antibodies that go 4 or 5 weeks after they birth. So this is really consistent with maternal antibodies. So perhaps they get antibodies through the sows and they clear up the antibodies in 5 weeks post-birth. A little bit of the epidemiology, as I did with PEDV. Look at this one. This is our case, weekly case. We keep track of cases, and it has, again, a pattern. And this is really interesting. I was looking at a report from the Producers Association back in the 60s, and they talk about this, let me remember, seasonal vesicular disease that goes through the summer. Of course, they didn't talk about PEDV. They just talk about seasonal vesicular disease, which is filled with this, with a seasonal pattern that we see now during the summer. So in summary, what we have seen for the last 2 years is an increment in vesicular disease and neonatal mortality, both associated with the presence of Seneca virus. We identified this new or contemporary strain, we like to call it, that is a little bit different from the historical strain of Seneca virus, excuse me. This disease is new for us. We're still learning a lot, and there's a lot of questions that need to be answered. So what is the origin of this viral mutation? We don't know if it is a genetic pressure or honestly, we don't know exactly. The host pathogen interaction, what is the molecular part of the interaction that we need to understand probably to prevent the disease. Predisposing factors, we know that virus, it can affect animals, as you see, and they don't get clinically sick. So probably they have a predisposing factor that make them sick. Clinical, serological, and viral prevalence, there are ongoing studies, so we're going to learn soon how widespread is the virus through the United States. Viral persistence in the environment and viral activation. I should probably cross this out. There's a really nice paper came out from University of Minnesota where they evaluate the environment persistence and the viral inactivation. So with that, I'm glad to take any of your questions. Thank you.
Questions for Dr. Pineyro?
For the sequencing of the PRRS virus, what percentage do we look at now, and how much more of a process is it to do the entire sequence?
So now with the OR5, so we look at the whole OR5, which probably is where we see the more variation. That is why we use that region. So now with this technology, the NGS, the next generation sequence, we can get the full length sequence in 3-4 days. That's the timeline. So, but let me take that back. 3-4 days to get the sequence. Then we need a few more days to get the analysis, which is a different part. So just the sequence itself, we can get it up to 3-4 days. Yep.
I might have missed it at the beginning, but did you have any opinions yet on the nanoparticle vaccines for PURRS?
For PURRS.
And any cautions that might go with trying those as they come to the market in terms of possible cautions?
Honestly, I know they are experimentally available, but I don't know if anyone have even tried to apply it in the market. And if they don't go through the market, we don't see it through the Diagnostic Lab. So I don't have much experience with that. Sorry.
Any other questions? Well, if there's no questions, let's thank Dr. Pineyro one more time.
Thank you.